Hello students, in the previous class we have discussed application of biotechnology in agriculture. In that the important application is production of GM crops or GM plants, that is genetically modified plants we have studied, and we have seen some of the examples of these GM plants, that is a production of insect resistant plants, for example BT cotton we have seen, and parasite nematode resistant plants we have seen, that is tobacco plants resistant to parasite nematodes by the technique of biotechnology, that is RNA interference. So these examples we have seen as part of application of biotechnology in agriculture. Today we will see application of biotechnology in medicine or medical biotechnology, right? In case of application of biotechnology in agriculture, we have seen based on what type of technique the crops were made resistant towards a group of insects. It is because of the ideology based on the toxins produced by Bacillus thuringiensis. These toxins are produced by certain genes of the plasmids of Bacillus thuringiensis known as cry genes, which code for cry proteins or crystal proteins, or they are known as delta endotoxins. Right? So that was the ideology behind the construction of genetically modified plants, that is a BT cotton plant, and based on RNA interference we have seen how the plants are resistant to parasite nematodes because of the main precursor for RNA interference is the complementary double stranded RNA, right? So today we will see the application of biotechnology in medicine, or how the medical biotechnology is evolving, like how the biotechnology is contributing in the medical field. The field of medical biotechnology is now experiencing rapid growth, okay, that is leading to the development of several innovative techniques. The techniques are used for preventing the diseases, for diagnosing the diseases at the molecular level, and they are used in the treatment of the diseases. So especially for treating a disease, how the biotechnology is contributing is it helps in the production of therapeutics. We have already studied it is helping in the large scale production of the therapeutics that are produced using genetic engineering techniques, that are produced using genetically engineered organisms, right? Not only the therapeutics are used, as well as the diagnosis techniques are also used, which we will be reading in further classes, studying in further classes regarding the techniques that are involved in the diagnosis. Before a disease could go to advanced stage, how we can diagnose that particular disease at the earlier stages. That also, once again, it is the biotechnology is helping to diagnose the diseases as well as to prevent the disease based on the techniques that are used to study the effects of the drugs on the disease. Okay, the prevention can be done if we know the root cause of the disease. So all these things can be achieved with the evolving development in the medical biotechnology. Let us see some of the therapeutics or medical biotechnology products that is helping in the treatment of the diseases. Examples include HGH, which is a human growth hormone. This is produced from E. coli cells via genetic engineering, and that is used for treatment of dwarfness. HGH, human growth hormone, helps in the treatment of dwarfness by expressing it in E. coli cells. Another example is chorionic gonadotrophin hormone, which is produced once again by recombinant DNA technology, and that is used for treatment of infertility, right? And interleukins are produced in E. coli cells via genetic engineering, and they are used for stimulating immunity system. Interleukins are used for stimulating immunity system, and platelet derived growth factors are produced by a recombinant DNA technology, are used for stimulating wound healing by clotting the blood, and genetically engineered insulin is also produced from E. coli cells that are used to treat diabetes, production of vaccines for influenza, virus, hepatitis A and B are also being produced using the biotech biotechnology techniques. Not only the vaccines, there are an n number of biotechnological products are being produced. Okay, one another example includes tissue plasminogen activator. It is an enzyme that is used to dissolve the blood clots after the heart attacks. Right, these are some of the examples of the biotechnology products that are being produced, and most of them are in use in the current scenario. Almost 30 recombinant therapeutics have been approved. Many are produced, many are researched and developed, but 30 recombinant therapeutics have been approved. Among them, only 12 are being marketed. Right now, only 12 are being marketed. So, among these recombinant therapeutics, among these biotechnological products, the one which is being marketed now is genetically engineered insulin, or it is known as recombinant insulin. So, in this class, we are going to see in detail about the recombinant insulin or genetically engineered insulin. What is the use of this? How it is produced in recombinant DNA technology? What is the function of this recombinant insulin? Right? So, you people might have heard about human insulin or humulin, which is genetically engineered insulin. There is a company in Bangalore, Biocon, which is a biopharmaceutical company which is going to produce this insulin. Okay, this insulin, first it was the hormone insulin first was discovered in the year 1921 by scientists Frederick Banting and Charles West. They have discovered this insulin in 1921 for the treatment of diabetes. For the treatment of diabetes. So, in our human body also, this insulin hormone is secreted. It is secreted by the beta cells of the pancreas, that is islets of Langerhans, which is going to regulate the blood glucose level. This is going to regulate the blood glucose level. Whenever there is increase in the glucose level, in order to regulate it, in order to monitor it and bring to the normal glucose level, insulin is being produced by the beta cells of the pancreas. So, how this insulin will be produced? For any protein to be produced, the basic thing is gene. A gene is going to be expressed, then only it is going to be replicated, transcripted and translated to form the protein. Okay, so even the insulin gene is present on chromosome 11 in our human genome. In our human genome, the gene coding for the insulin is present on the top of the chromosome 11. Okay, so if in case of diabetic patients, what will happen? They, due to the damaged beta cells or due to the non-functional production of the insulin, or in case of the ability of the body not to use the insulin, the body glucose level is going to be increased, right? That is leading to the diabetic condition. In order to regulate the glucose level, the body has to produce the insulin. But in case of diabetic patients, the body doesn't have the proper cells to produce the active insulin. Therefore, the insulin must be supplied to the body externally. The insulin must be supplied to the body externally. So, in earlier days, in order to treat this insulin, what was happening? Because the body, in order to meet the requirement of this external supplying of this insulin, what will happen? We need a large requirement or large production of the insulin. The body cannot produce very large content, especially it is unable to produce to meet the requirement of the body itself. So to extract large amount of insulin, people used to extract this insulin from pigs and cattle. Even the pig and cattle contains the gene that codes for the insulin, and they are almost similar to that of the human beings, right? So the insulin that was extracted from the pig, it is known as porcine insulin, and the one which is extracted from cattle, it is known as bovine insulin, right? In order, in the earlier days, to treat this diabetes, the insulin that was extracted from the pig, that is porcine, and the one that is extracted from cattle, that is bovine, is being supplied to the patients. But there was a drawback. Okay, though the insulin that was similar, okay, to that of the human insulin, but there is a slight modification in the number of amino acids. The bovine insulin was varying with the human insulin with 2 amino acids, whereas the porcine insulin was varying with the human insulin with 1 amino acid. Okay, both they are similar in human insulin, but their composition is slightly different. Therefore, repeated use of this foreign insulin in the human body was resulting into the adverse effects, and it was creating swelling, allergic reactions were produced. So therefore, that made a way, or that made a challenge to produce insulin via recombinant DNA technology. That made a way to produce insulin via recombinant DNA technology. Before we could move on how to produce insulin via recombinant DNA technology, let us see first what is the structure of insulin. What is the structure of insulin? So in the human body, as well as in case of other mammals, the insulin is produced as a proinsulin. It is produced as a proinsulin, which means it is a inactive insulin, right? First, it will be produced as a pre-proinsulin. Pre-proinsulin, which contains a signal peptide, will be present. Okay, this signal peptide will be cleaved and it forms a proinsulin, and then once again the proinsulin, further some polypeptide chains will be removed, and it results in the formation of the active insulin. So what is the structure of this active insulin? It is made up of 2 polypeptide chains. The active insulin is made up of 2 polypeptide chains, that is chain A and chain B, right? Chain A contains 21 amino acids and chain B contains 30 amino acids. Getting, students? The structure of insulin. Active insulin contains 51 amino acids, which is made up of 2 polypeptide chains. Chain A is made up of 21 amino acids and chain B is made up of 30 amino acids. Chain A and chain B are linked by disulfide bridges or disulfide bonds, right? In animals as well as including humans, this is the structure of the active insulin. Chain A is made up of 21 amino acids and chain B is 30 amino acids. Chain A and chain B are linked by disulfide bonds, right? Now, in most cases, that's what, in humans as well as other mammals, it is produced as a proinsulin or a inactive insulin. So what this proinsulin is containing? Proinsulin contains chain A and chain B, which is attached to a extra stretch of C peptide. Getting? There is an extra polypeptide chain that is connecting the chain A and chain B, which is known as C peptide, and this C peptide contains 31 amino acids. It contains 31 amino acids, right? Getting, students? Usually, initially, it is formed in the human body as well in other mammals as proinsulin, which is inactive insulin, and this inactive insulin contains chain A and chain B linked with chain C. Chain C is going to link chain A and chain B, and this chain C is containing 31 amino acid. After the cleavage of this chain C by the peptidase enzyme, okay, the chain A and chain B are linked with the disulfide bridges and release the C peptide or chain C, which is known as maturation of proinsulin to active insulin. Getting proinsulin contains chain A, chain B and chain C, whereas active insulin contains only chain A and chain B. Here you can see the maturation of proinsulin to active insulin. So here in this diagram, the proinsulin contains 3 polypeptide chains, chain A, chain B and chain C. Chain A is represented by blue color, chain B is represented by green color, chain C is represented by grey color. Getting students? Here chain C or C peptide is connecting chain A and chain B, is linking the chain A and chain B. Chain A is made up of 21 amino acids, chain B with 30 amino acids and chain C with 31 amino acids. So what will happen during the maturation process? Ribosomes are the sites for the protein synthesis. So at the rough endoplasmic reticulum, this is going to be formed. The proinsulin is formed and later in the Golgi, it is transferred to the Golgi body as a secretory vesicle, and then here it is going to contain the extra stretch C peptide is present, that is removed by the Golgi body and it is packaged to the destination, to the required cells when there is a requirement. The whole chain A, chain B linked with the disulfide bonds and the free C peptide is sent to the required destination cells. Right? Now chain C or the free C peptide is going to remain dysfunctional because it doesn't have any function, whereas chain A and chain B that are linked with disulfide bridges is going to have a function in regulating the blood glucose level. So this is maturation of proinsulin to active insulin. Now let us see how this insulin is genetically engineered or how this insulin is produced using recombinant DNA technology. Right? In 1979, first the insulin was produced by recombinant DNA technology. Okay, in the E. coli cells it is produced. The human gene that is coding for insulin was isolated from the animal cells and then it is inserted in the vector and then it is introduced into the bacterial cells and made these bacterial cells to express this gene. But there was a problem in expressing this gene, that is maturation of proinsulin to insulin was a great challenge that was faced when it is produced in the year 1979. Okay, the maturation of proinsulin to insulin was unable to be produced in case of first trial in the year 1979. Later, in the year 1983, there was a company, American company known as Eli Lilly, a US company that has produced insulin via recombinant DNA technology, okay, that was approved for human use, right? Even in 1979, that also produce the insulin, but there was a problem in maturation of proinsulin to insulin, and that was not approved for human use. Whereas in the year after 1979, there was lot of research has taken place in finding out the best way to produce insulin through recombinant DNA technology, and later in the year 1983, Eli Lilly American company produced insulin that was approved for human use. How they have produced the insulin via recombinant DNA technology? If we are isolating the whole gene from human cells and it is allowed to express in the bacterial expression system, there was a problem in maturation of proinsulin to insulin. That is the reason they have now the structure of insulin shows that it is made up of chain A and chain B, and they are linked with the disulfide bridges. So taking into the technology or the ideology of the structure of insulin, the synthetic genes coding for chain A and chain B are synthesized, means artificially in vitro in the laboratory. The chain A is synthesized separately and the chain B is synthesized separately. The genetic information was taken. What is the composition of the chain A? It is made up of what type of nucleotide sequence, and the genetic composition of chain B is known. Okay, based on that information, synthetically the chain A and chain B are synthesized separately in the laboratory, right? And then the genes coding for chain A is introduced into the vector. Right now, how this recombinant DNA technology will take place? First step is isolation of the genetic material or isolation of the desired gene. Okay, since from the source they were unable to take up the gene because they were facing the problem in the maturation of proinsulin to insulin, so the synthetic genes were produced. So we have first step is done. Okay, the desired genes are being isolated, that is the synthetic genes are produced that codes for chain A and chain B. Second step is what? Cloning of the genes. How these genes can be cloned? They are first linked to the vector DNA. So the vector used is PBR322, which is a artificial or genetically engineered plasmid vector that is suitable for cloning genes in the E. coli cells. Right? Now the chain A, the gene coding for chain A is linked to the vector PBR322, a different vector, and chain B, the genes coding for chain B are once again introduced into the another set of PBR322 vectors separately, and these are introduced into the E. coli cells separately. Means in the same E. coli cell they are not introduced. In different E. coli cells chain A is introduced, and different E. coli cells carrying the vector PBR322 chain genes coding for chain B is introduced. Right? After the cloning process, the next step is what? Introducing this into the E. coli cells. Right? And expressing this in the E. coli cells. So in case of large scale production, what will happen? First, in the R&D level, screening will take place. After the E. coli cells are made competent, the vector PBR322 containing the genes that is coding for chain A separately and chain B separately are introduced in the E. coli cells using the gene transfer technique. Now you must know which type of gene transfer technique or how these E. coli cells are made competent. There is a process that is a heat shock method. Using that, the E. coli cells are made competent to take up the vector PBR322 containing chain A separately and chain B separately. Right? And the next step is obtaining the foreign gene product. Right? This is one of the step of the recombinant DNA technology. Obtaining the foreign gene product, that is growing them in large scale. So first comes the upstream processing will take place, then comes the downstream processing is going to takes place. So growing these E. coli cells that is containing the recombinant DNA in a specific required nutrient medium so that the genes coding for chain A will express and form the chain A polypeptide, and the genes coding for chain B are going to express and form the chain B polypeptide. Right? Okay, then after the upstream processing, okay, then comes the purification. So in the upstream processing, E. coli cells will absorb the nutrients present in the nutrient medium, and they grow in size, and they multiply, and their genes are expressed, and the product is formed. Now the product formed is within the cell, so next process it has to undergo is downstream processing, which has to be performed separately for the cells containing chain A and separately for the cells containing chain B. So downstream processing involves centrifugation process, filtration process, that is segregation of the cells in case of the extracellular product. If it is an intracellular products, then it requires cell disruption or cell lysis, and then filtration process. Then purification steps are done, which involves chromatographic techniques are involved to contain the product in the purified form. Right? Now chain A and chain B are in a purified form, but our insulin structure is the chain A and chain B are linked with the disulfide bridges, or they contain the disulfide bonds. Now to develop these disulfide bridges, the chain A and chain B that are extracted separately are both mixed together using sodium sulfonate and sodium sulfite. They are mixed using chemicals sodium disulfonate and sodium sulfite, that is going to form the active human insulin. Clear, students? That is going to form the active human insulin. This human insulin is going to, it is given through injections only, but not through the pills or the tablets. Why? Because now each enzyme and each hormone has its own optimum pH where it is going to remain active. So insulin is going to remain active at the pH 7.6 to 8. It is going to remain active in the pH 7.6 to 8. If the tablet or the pills are being produced, when it is taken orally, what happens in the stomach? There is the acidic pH present, because of which it gets degraded. So the research is still going on to develop the insulin tablets, whereas time being now the insulin is produced and it is injected to the which is going to be directly dissolved in the blood, and it is going to control the blood glucose level. So this is here, it is a recombinant DNA technology. You can see there is a promoter gene is present. Okay, you have a vector here. Left hand side the diagram shows chain A, right hand side diagram shows chain B. Right, students? Here this is the plasmid vector PBR322, and here it is plasmid vector PBR322. Now this plasmid vector PBR322 contains antibiotic resistant gene acting as a selectable marker, and the promoter gene is present, promoter gene which regulates the gene expression, and it also decides the location where the gene product is formed. So the promoter gene is present near the gene coding for the enzyme beta galactosidase. This enzyme is going to convert the lactose into glucose and galactose, right? And insulin gene A subunit is present, means this gene is coding for chain A insulin. The chain A insulin is coded by the insulin gene A. To form the chain A polypeptide, okay, gene coding for insulin is represented with a red color. Now this gene is placed near the promoter and near the beta galactosidase gene, because when the beta galactosidase enzyme is expressed, along with that chain A polypeptide chain is also produced. Clear? So after the recombinant DNA, that is vector with the gene of interest, it is inserted into the E. coli cell using the gene transfer technique. Using the gene transfer technique, it is inserted into the bacterial cells here. Chain A separately and chain B separately. Now, in these bacterial cells, the plasmid is here. Okay. After the growth of the plasmid in the upstream processing, what will happen? It is going to produce the product. Okay. Here, the red color represents the chain A polypeptide, yellow color represents the enzyme beta galactosidase. Okay, so together it is known as a fusion protein. Now, next step is what? You have to separate this beta galactosidase enzyme from the chain A insulin, and here also beta galactosidase from chain B polypeptide. This can be segregated via a chemical known as cyanogen bromide. A chemical cyanogen bromide is used that is going to segregate the enzyme from the polypeptide chain. So cyanogen bromide is used. Now we have chain A separately and chain B separately that has undergone the downstream processing, and now these two purified chain A and chain B are mixed with sodium disulfonate and sodium sulfite to form the disulfide bridges. They are mixed with sodium disulfonate and sodium sulfite to form the disulfide bridges, that is resulting into the active insulin. You can see here, this is the active insulin which is packaged and marketed. Clear, students? That is packaged and marketed. So this is how genetically engineered insulin is produced, okay, using the biotechnology techniques. Now this insulin is used worldwide to control the diabetes. So this is one of the important product of the biotechnology that has application in medical field, which is used as a therapeutic to treat diabetes, to control diabetes.